Welcome to Unboxing Biology with Smriti. In the previous video, we have discussed about test cross, back cross, and the reciprocal cross. In this video, we will be discussing about incomplete dominance. Okay, so this incomplete dominance is the variation from the Mendel's laws or the exceptions from the, it is dominance. So it is exception from the Mendel's law. Fine. Before knowing incomplete dominance, I will tell you some term. First is locus. So, locus is defined as the position of a gene on the chromosome. Okay? So, let us learn it with a diagram. Suppose we are taking here the homologous pair of chromosomes. Now, here on chromosome, here is a particular gene present on the chromosome. Suppose it is responsible for the character of the stem length, that is tall. It can be here also tall. Fine. It can be also the dominant form of stem length. Fine. Now, if the interaction is done between these two alleles, okay, so these two alleles, if it is interacting and giving any type of phenotype, then this type of interaction is known as intragenic. Fine. So now this is intragenic. Locus, you can say here the position of a gene on the chromosome, and intragenic means when two alleles, or it can also be written as, or we can write it as intraallelic. Fine. Now let us take another pair of chromosome. Now, okay, so we are taking here the two chromosomes, two homologous pair of chromosomes. Now, here, let us take here again the same example. It is here a gene is present which is responsible for the character of the stem length. Fine. Here, again here the gene is present, but now here the gene is present for the stem, or the flower color. So flower color is violet and white. Fine. Now, if one gene from here is interacting with the another gene present on the another chromosome, or it can be between the same chromosomes, if the one gene is interacting with the another gene, it is the term given is as intergenic. Fine. Now, intra means within the genes and inter means between the genes. Fine. Now, we have to study both type of interaction. In intragenic, we are going to study about the incomplete dominance and co-dominance. Okay. Whereas in the intergenic, we are going to study about the polygenic inheritance. And epistasis. Fine. So now, at first we will study the intragenic type of interaction, that is the example we are taking here, the incomplete dominance. Fine. So in incomplete dominance, it means with the term only you can understand. It is first let me tell you about law of dominance. If you remember, Mendel told that whenever we start with pure lines, it is the dominant which expresses itself. Fine. Now, Incomplete dominance is nothing but actually the exception case of the law of dominance. Exceptional case. That is, here the dominant gene is not able to completely express itself. It is expressing but incompletely. So when they are incompletely expressing, the intermediate type of phenotype is expressed, which is different from the parents. Fine. Now we cannot tell that blending is happening. Why? I will tell you with examples. Blending is not happening here because in the F2 generations, the parents are again recovered, or you can say again appears. Fine. So parents are again appearing over here in the F2 generation. Fine. Now that's why you can say that blending is not happening here. You cannot use the word or term blending. Now we will start with examples. Okay. So it is important to learn examples. First example which is showing the incomplete dominance is Antirrhinum majus. Okay, whose common name is dog flower. Okay, now the second example we are going to take is 4 o'clock plant. Now this 4 o'clock plant is a common name, but the scientific name here is Mirabilis jalapa. Fine. So I'll write the name Mirabilis jalapa. Fine. Common name is 4 o'clock plant and its scientific name is Mirabilis jalapa. Now the third example which shows the incomplete dominance is a bird named as Andalusian fowl. Fine. I'll tell you these two, we are dealing here with the flower color. Okay. I'll tell you the examples or the character taking the flower color of these two plants. Fine. But in this Andalusian fowl, we will take the character color of the feather. Fine. So we will start with Mirabilis jalapa. Both Mirabilis jalapa and the Antirrhinum majus shows the same kind of color of the flower, that is red as dominant and white as the recessive. Fine. So we are taking here the Mirabilis jalapa as the example. Now both type of flowers produce red and white. Red as a dominant, whereas small r as white. Here it is recessive, here it is dominant. Okay. Now these are the parents. You can write over here as a male and a female. Wait. Okay, so this is male. Fine. Now what do we have to do? We have to produce or take out the gametes. So we are taking here the gametes out of the parents. Okay. So the gametes will be capital R, small r, and again small r. Okay. So now after taking out the gametes, we have to establish the Punnett square for F1 generation. Okay. So let me make a quick Punnett square. Okay. Again, the two types of gametes are produced over here. That's why we are making two columns in two rows. Okay. So now here this is expressing the F1 generation. Fine. We will write it here F1 generation. Okay. Now take it here as a male, here as female. Now we will write the gametes over here, capital R, capital R. This is a small r, small r. Now the offspring which is produced is capital R, small r, capital R, small r. Again same and again the same. Fine. So now, if in the case of law of dominance, if we start with pure lines, which we started over here also. Now, if we start with the pure lines, it is a dominant character or the dominant one which expresses itself, okay? But in this case, here it is capital R, small r, that is the dominant and the recessive. But here the dominant is unable to completely express itself. It means this flower color or the offspring color will not be red in color. It will be pink in color. Because the dominant here is not able to completely express itself. Now all the four type of offspring will be pink in color. Fine. Now we will write the phenotypic ratio and the genotypic ratio. Here the phenotypic ratio will be 4:0, where the 4 will be pink flower, whereas 0 will be white, or you can say red. Now, in the genotypic ratio, again it will be 4:0, as 4 of them are heterozygous in nature. Okay, so now we will do selfing of this and this offspring. Okay, now selfing means the self fertilization. Here we're doing selfing. So these are now here acting as the parents. Okay, now we have to take out the gametes of the parents. Okay, so it will be capital R, small r, capital R, small r. Fine. Now we will now make the Punnett square to take out the F2 generation. Fine. So again, because there are two gametes, we are making the two columns and the two rows. Fine. Now here we will write the gametes. Okay. So let us take this as a male and a female. You can write it over here also. Now the offspring which is produced is capital R, capital R, capital R, small r, capital R, small r, and small r, small r. Fine. So this is the F2 generation. Fine. Now in the F2 generation, we again have to write the phenotypic ratio and the genotypic ratio. Fine. So the phenotypic ratio here will be, this is red as this is pure. Sorry. This is pink in color. This flower is again pink in color, whereas this is white in color. Fine. So the phenotypic ratio here will be 1, which is red, 2, which is pink, again 1, which is white. Fine. Now the genotypic ratio here will be 1 is to 2 is to 1, where 1 is homozygous dominant, another one is heterozygous. Fine, pink, and the third one is homozygous recessive one. So this is homozygous recessive. This is homozygous dominant. So if you remember the law of dominance, the phenotypic ratio which was produced in the F2 generation was 3:1. But here in the case of the incomplete dominance, we are getting the phenotypic ratio as 1:2:1. Fine. Now we will take another example, that was Andalusian fowl. Okay, so now this fowl, we are taking the character here is feather color. Fine. So now it produces two types of colors of the feather, black and white. So it is not totally white, it is somewhat grayish white in color, but we will write here white. So this will be small b, here it will be capital B, capital B. Again, this will act as parents over here. Let us write it as a male, this as a female. Now, the gametes which will be produced will be capital B, capital B. This will be small b and small b. So these are the gametes. Now, we have to establish the Punnett square, which will be representing the F1 generation of the above crossing. Fine. So, write this as a male, this as a female. Now, capital B, capital B. This will be small b, small b. Now, the crossing will be capital B, small b, capital B, small b, capital B, small b. Again, the same. This is the F1 generation. Now, again, if we see the law of dominance, it is the dominant character. When we are taking with the pure lines, we are starting with the pure lines, we get the dominant which expresses itself. But in this case, in the case of the incomplete dominance, it is not able, the dominant one is not able to completely express itself. That's why here it will be not black but blue in color. All the four of the offspring will show blue feathers. Fine. So now again we will write the phenotypic ratio. The phenotypic ratio will be 4:0, where if 4 is blue. Now the genotypic ratio will be again 4:0 because 4 of them are heterozygous in nature. Fine. Now we will again do the selfing thing, that is the self-fertilization. Okay. Now these are the parents. This is the male, this is female. Now we have to write the gametes. Gametes will be capital B, small b. Here also the same, capital B and small b. Now we will make a Punnett square which will be representing the F2 generation. Okay. Now we just have to write is a male and a female. Now capital B, small b, capital B, small b. Fine. Now the offspring will be capital B, capital B, capital B, small b, capital B, small b, and small b, small b. Fine. Now here it is the pure dominant. That's why it will show black feathers. Here it is heterozygous. Here again the dominant one is incompletely expressing itself. That's why it will show the color blue, again blue, and here it will be white as it is pure. So here the phenotypic ratio is 1:2:1, where 1 is black, against 2 is blue, and 1 is white. The genotypic ratio will be 1:2:1, where 1 is homozygous dominant, this one, 2 is heterozygous, blue, and the again 1 is here homozygous recessive, this one. Okay, so we will revise. Now, here it is the incomplete dominance we are studying, which means according to the law of Mendel, it is when we start with the pure lines, it is the dominant which expresses itself. But incomplete dominance is the exceptional case of the Mendel's principles. Fine. So, we have started with the term locus, which means the position of a gene on the chromosome. So, here we understood the intragenic, which means the interaction between the genes. Fine, between the genes. Again, within the genes or between the two genes, if the interaction is happening, it is intergenic. Inter means between, intra means within. So here, within the alleles or within the genes, the interaction is happening. Here, between the genes, interaction is happening. Okay, now we started the law of dominance, where we understood the law of dominance which was given by Mendel. According to him, if we start with the pure line, it is the dominant which expresses itself. But now in the case of incomplete dominance, here it is unable to express itself completely, so there is a production of the intermediate phenotype. Now we cannot use the term here blending. Why? Because in F2 generation here parents are again appearing. Fine. So now there are 3 examples which we have to remember. Antirrhinum majus, which is also known as, the common name is dog flower. 4 o'clock plant, that is the common name, and the scientific name is Mirabilis jalapa. And third is the Andalusian fowl. Here, now in these 2 examples, Antirrhinum majus and the 4 o'clock plant of the Mirabilis jalapa, we are taking the character flower color. But in the Andalusian fowl, we are taking the feather color as the character. Fine. Now we have took this example Mirabilis jalapa, and you can also write it over here as Antirrhinum majus, because both shows the same flower color, that is a red and white. Fine. So now this, I forgot to tell you that Carl Correns did experiment on Mirabilis jalapa and he gave incomplete dominance. Fine. Now here it is a red and white parents. The gametes are here, and again Punnett square was made, F1 generation was made, but here the dominant one was incompletely expressing itself. That's why it was showing pink flowers. Fine. Now in the F2 generation, why we are not using the term blending? Because in the F2 generation you can see that the parents are reappearing here. So the homozygous dominant and the homozygous recessive one. Fine. Now we have took the another example, Andalusian fowl, the feathered color as the character, which is showing the black and white. Now we did crossing between them and we got the blue feathers. What we have to get? That was black color according to the law of dominance, but we are getting here the blue color. Okay, so now again we did selfing and we got the phenotypic ratio as 1:2:1 and genotypic ratio as 1:2:1. According to the law of dominance, we were getting previously the ratio as 3:1, but here we are not getting 3:1. It is 1:2:1. Fine. So in the next video, we will be discussing about the co-dominance, which is again the type of intragenic interaction. Thank you for watching my video.